All right, translation. Morning. It's hard to understand transcription if we don't follow it up right away with translation. So we have a final mRNA transcript coming out from the nucleus, then translation can now commence. Let's look at it happening first before we discuss the steps. All right, so you have your DNA. This is inside your nucleus, which is transcribed into an mRNA. And this mRNA transcript, with the use of tRNA, is translated into a particular protein that you need. So this protein is brought to you by the code that you have here in the DNA. Okay. Now, how does it happen? So this mRNA coming from your nucleus will bind to the small ribosomal subunit. As it binds, the first tRNA comes in and attaches itself to the first codon. So always, the start codon is your, what's the sequence? AUG. So pretend this was AUG, so the first tRNA carrying its amino acid binds to that first codon. On the P site, there's 3 sites in your ribosomes, EPA, so it binds on the P site. Then it will translocate and read the next codon. The incoming tRNA binds to what site? The A site. Okay. And those two tRNA side by side, these 2 amino acids can now start to form a bond. What's the bond between 2 amino acids? Peptide bond. What's the process? Condensation, hydrolysis? Condensation. So a reaction will occur there. Those 2 amino acids will start to form a peptide bond. We know that already. There. And the first tRNA is now released as it moves to the E site. So you can think of the E site as exit. So it's released. And then as you keep translocating, as you keep reading the mRNA codon per codon, another tRNA binds again to the A site, and then a peptide bond is formed again in your 2 amino acids. It moves and it leaves. So on and so forth until you reach your stop codon. Okay, we'll learn about the stop codon. The stop codon is either, reading notes, what are the stop codons? Yeah, what's the codon? UAA, UGA, and UAG. Yes, correct. So if it reaches either one of those 3 stop codons, translation stops. And what is your final product? You have a polypeptide. This is just a chain. And in your reading notes, this chain will be folded into your secondary structure, more folding into your tertiary structure, combinations of your tertiary and secondary, you have your quaternary proteins. Okay? So in other words, 7.3 is how to make proteins using your DNA code. All right, so let's begin. First concept you need to know is your tRNA activating enzyme. This is an enzyme found in the cytoplasm, and a funny way to remember this is the TAE, tRNA activating enzyme. It kind of looks like poop right there. If you're Filipino, you understand. If not, ignore me. So this enzyme right here is in charge of binding the specific amino acid to a specific tRNA. Logic question. If you have 20 amino acids, then how many types of tRNA do you have? If you have 20 specific amino acids, then how many tRNAs do we have? 20 also. There's 20 types of tRNA, each tRNA binding to a specific amino acid. And each tRNA that is specific to a specific amino acid is using 20 different enzymes as well. Do you follow? Enzymes are specific, right? So in this particular example, this TAE is specific for binding valine to your tRNA. So how does this happen? We need ATP. So not only do we need ATP for muscle contraction, you might think, oh, you need ATP for energy. This is one example of the use of that ATP as a source of energy. So what happens? ATP will bind with two low. Your enzyme has two sites. One site for the amino acid, one site for your tRNA. So your ATP will bind together with a specific amino acid, so valine right here. The phosphate, two phosphates will bind to your amino acid. Look at the product here, AMP instead of ATP. What happened to the two phosphate? It bind with the amino acid. So if your amino acid here, it's now activated. Binding of that binding of that ANP from ATP activates your amino acid, and then it will signal the binding of your specific tRNA. In this case, valine is coded by CAA. I don't know, double check codon table, please. So this particular tRNA binds to another site in your enzyme, and this enzyme closes the gap, binds that amino acid to the tip of your tRNA. What is your final product? Your activated tRNA. It's a tRNA containing an amino acid. So you can have tRNA floating around in the cytoplasm but not attached to an amino acid. It needs to be activated first by your tRNA activating enzyme. Question. I forgot the answer, so I'm asking you. If you read your notes, where does the tRNA, where does the TAE put the amino acid? At the 5' end or at the 3' end? Yeah. Is it not in your reading notes? It's orange. So what is it? Can you please read? I actually forgot the answer. What does it say? Okay. Thank you, Alexa. Please forever remember that. Amino acid is attached to the 3' end of your tRNA. Please don't forget that your tRNA is also a nucleic acid. It's also an RNA. It looks like your mRNAs, but it's folded. Look at the folding. If I unfold that, that's one long RNA strand. But it's folded in such a way that it forms 4 corners. So this, loop, and then the end there. So it's also from 5' all the way to 3'. So we add your amino acid at the 3' end. There's a commercial story on Facebook. There was a trending video about this preacher. He was like, if you look at your cells molecularly to the smallest level, the enzyme responsible for making your proteins is a cross shape. I'm like, sure, yeah, it is a cross shape. I mean, yeah, wonderful to know. But it's not because it's Jesus Christ. It's just wonderful to know, like, it looks like a cross. And it helps you express your genes. There was another molecule, hold on. Cross-shaped molecule. Laminin. This one. This was another cross-shaped molecule, laminin. And laminin is a membrane protein that is responsible for cell adhesion. So how can cells stick together? They have laminin. Wow. Another cross. Maybe the DNA is cross-shaped too. This one is the electron micrograph of laminin. Kind of looks like an X. Kind of looks like a cross. Okay, enough of your commercial. So Alexis said the tRNA activating enzyme adds your amino acid at the 3' end of your tRNA. Now, stages of translation. Quick review. Initiation. The first tRNA looks for the start codon, which is AUG. Okay. And as it attaches to the first codon, it positions itself, actually in some animation, this tRNA needs to bind to the AUG first before the large ribosomal subunit attaches. Okay? It's some animation, the textbook also said that. So this needs to attach first before your large ribosome subunit comes in. If it does, the tRNA is located in the P site of the large ribosomal subunit. What happens? So mRNA binds to mRNA binds to your small ribosomal subunit. It locates the AUG codon. After binding, the large ribosomal subunit comes in, and the tRNA is now situated in your P site. Okay. Next, elongation is just translating your mRNA code one codon at a time, as we have seen in the animation. So it translocate. Translocate means reading codon per codon, 3 bases at a time. And then termination, if it reaches, oh, sorry, nobody's telling me it's zoomed in. You're not listening to me anymore, like daydreaming already. I'm like in your brain, I'm like, ooh. It happened to me one time, English, my teacher, I was so tired, so I did like this. I think I told you the story. And then she kept talking, da 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 da. So I closed my eyes because I'm just tired. And then when I opened my eyes, I'm like, where am I? I was in deep sleep already. Where am I? Ooh, I'm in my English class. I fell asleep. So thanks for daydreaming. All right, termination. Either you reach Filipinos, it's easy to remember. Wag, don't, uga, shake, or else the baby will cry. Uwa. Wag, uga, uwa. If you don't get it, just memorize it, please. Wag, uga, uwa. The baby will cry. All right, if it reaches that sequence, another protein will bind instead of your tRNA. That's called the release factor, signaling the termination of translation. And this is your growing polypeptide chain. All right. Where does it happen? Cytoplasm. All right, let's make it a little bit hard. There's 2 types of ribosomes, free ribosome and RER. So the ribosomes in the cytoplasm make proteins that are destined to be used inside the cell. The ribosomes in the RER makes proteins that are destined to be packaged and sent out of the cell. Okay? Now how does that happen? How does the mRNA know whether it's going to be translated in the free ribosome or in the RER? Let's look at an animation.
All protein synthesis in eukaryotes begins in the cytosol, where ribosomal subunits come together and begin translating an mRNA near its 5' end. Soon after the protein starts to emerge from the ribosome, it may become directed to the endoplasmic reticulum, or ER. It is targeted to the ER if it is ultimately destined for incorporation into the ER, the Golgi apparatus, lysosomes, or plasma membrane, or if it is destined for secretion. In mammalian cells, most proteins enter the ER co-translationally. These proteins have an amino acid sequence called a signal sequence, which is typically located at the amino terminus of the polypeptide chain. Signal sequences span about 20 amino acids and include a stretch of hydrophobic amino acids preceded by a basic amino acid. As a signal sequence emerges from the ribosome, it is recognized and bound by a signal recognition particle, or SRP, consisting of 6 polypeptides and a small cytoplasmic RNA. The SRP binds the ribosome as well as the signal sequence, and in so doing, stalls translation. The SRP targets the entire complex to the rough endoplasmic reticulum by binding to an SRP receptor on the membrane of the ER. GTP molecules bind to the SRP and the SRP receptor. The GTP binding triggers the transfer of the signal sequence from the SRP.
Okay, too long. Let me summarize. Imagine all of your translations all of those translation of your mRNA transcripts, all of them happen in the free ribosome. All. But if your sequence of transcription, if your mRNA has a sequence called a, what was it? Signal sequence. SERPs, SRPs, will recognize that sequence and will drag your translation into the endoplasmic reticulum. Now it becomes a bound ribosome, or we call RER. So those free ribosomes can actually attach themselves in the endoplasmic reticulum if there's a signal sequence for the protein to be translated um, in the endoplasmic reticulum. So this one will drag this transcript into the membrane of your endoplasmic reticulum, and translation can keep continuing there, so that your final protein is found where? Your final protein is found where? Inside the endoplasmic reticulum. Why does it need to be inside? To be packaged into a vesicle and sent to the. Where does it go after endoplasmic reticulum? No, what organ? Golgi. From ER to Golgi. Golgi finishes the folding of your protein. Golgi sends a vesicle to be released by the cell. So we're going back to your organelles now. That's the reason why Golgi packages proteins, because it's getting the vesicle from the ER. And how does the vesicle start here? Because you translated in the ER. And why did you translate in the ER? Because it has a signal sequence that signals your molecules like, hey, don't translate here in the cytoplasm, but translate it here in the endoplasmic reticulum. Easy concept to understand. Don't worry about the jargons, just please know the word signal sequence. Signal sequence, arrow, endoplasmic reticulum. I'll end with 2 slides, HLs. So this is your, what is this molecule? Yeah, so thank you, Alexa. What is my pink strand? What is that? That's your mRNA. By the way, don't think that the small ribosomal subunit is always in the bottom. It can be on the top or on the side. Doesn't matter. So that's your mRNA. This is your growing polypeptide chain. But then it has a signal sequence. So your SERPs will drag those ribosome into your endoplasmic reticulum membrane. And translation can keep continuing until your final product is now located inside your endoplasmic reticulum. So this is why BioHL is fun. Like, this is a really fun concept. That's not basic. Wow, you know something complex. I'm so proud of you right now. Where will this go? It will form into a vesicle, right? This will merge with the membrane and form a vesicle. It will go to the Golgi. So like this. It will form a vesicle. It will pinch off as a vesicle shipped to your Golgi. For packaging. Because this is not folded final. We still need to fold it into secondary, tertiary, or quaternary proteins. Don't move yet. Don't move. Are we clear with this? This is not in the old curriculum, so I'm kind of emphasizing free ribosomes and ribosomes in your ER. So free ribosomes, it's destined to be used inside the cell. Those that needs to go to the plasma membrane or released through exocytosis or used by your lysosome or endosome, it's made inside your endoplasmic reticulum. Can we, like, before I let you go, I want you to, what do I do? Like, think, talk to each other. Please explain the difference between bound ribosomes and free ribosomes. Explain it to each other, just verbally process it.